Hello, everyone, and welcome to Best Practices for Clinical Labs in Turbulent Times. I'm Cassie Soltman of LabRoots, and I'll be your MC for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now pass it over to Mara Aspinall for an introduction on the webinar.
Hello, everyone, and welcome. Welcome to Best Practices for Clinical Labs in Turbulent Times. If you've been paying attention to the news, you know that that title is very important. It is definitely turbulent times. Just earlier this week, a federal court in Texas vacated the FDA's final rule for laboratory developed tests. But that doesn't mean there are no best practices for clinical labs. So we will go forward with our webinar today and truly talk about what are the best practices in the US and a bit around the world to create the highest quality, most effective lab. With that, let me start by acknowledging Thermo Fisher Scientific. They have provided funding for this webinar. You can see the disclaimer on the screen. As I say hello, I can tell you that my name is Mara Aspinall. I'm the co-founder of the ASU School of Biomedical Diagnostics. More on that in a few minutes. I'm a partner at Illumina Ventures, where we invest in genomic companies. I have 35 years industry experience. I have seen a lot of regulations come and go, and therefore can really talk about the importance of this moment in our industry. With that, I am very committed to education, and the ASU program is unique. We offer a master's degree in biomedical diagnostics. You can see some of the information here on the screen, and those who are interested, please follow up with us. But today, again, is about the best practices. How can you build and continue to improve the quality and the functioning of your clinical lab? With that, we have two great speakers, both from Arizona State University. First, you will hear from Michael Donovan. He is the program director and assistant teaching professor in biomedical diagnostics at ASU. He is an attorney by training, has a lot of experience in life sciences broadly, particularly in patent prosecution and intellectual property. Our second speaker is Travis Schlappe. Travis is an assistant teaching professor in biomedical diagnostics. He came to ASU from extensive experience at the Keck Graduate Institute and has a lot of experience in innovation and the business of diagnostics. So with that, I'm very happy to pass it over to Michael Donovan to discuss our learning objectives, agenda, and then begin our webinar.
Thank you so much, Mara. Good morning, good afternoon, good evening, everyone. We know we've got people from all over the world on this webinar. Thank you for joining us. We appreciate you taking part of your day to learn a little bit about some of these best practices. So from a learning objective standpoint, you should be able, by the end of this talk, you should be able to evaluate these quality system considerations. As we'll talk about both in a few minutes and throughout much of this discussion, these ISO standards, depending on particular situations, may be aspirational. They don't necessarily, you may not necessarily need to comply from a regulatory standpoint, but these are best practices. These are ways that you can improve the operations of your clinical laboratory and provide safe and effective results for patients. In addition, by the end, we hope you're able to assess these strategic responses for these labs and develop a strategic plan to stay in compliance. Even though the LDT rule, and I'll briefly touch on that in a few slides, is looking like it's not going to come into effect on May 6th, but as I said, these are still best practices that can help your laboratory operate in an efficient and a safe manner. So in terms of what we are going to go over, we're going to do just a very brief introduction, talk a little bit about our timeline in terms of compliance, or in terms of the U.S. government's attempts to regulate LDTs. And then we're really going to focus in on those quality systems in clinical laboratories. And then we'll wrap up with a little question and answer session. And any questions that you may have, please put them in the Q&A box. We'll get to as many as we can, and those that we don't get to, we will respond to after the fact. All right, so where do we stand today in terms of these best practices? There are a few different ISO standards that are trying to drive quality in the clinical laboratory setting. We have ISO 15189, which is the kind of the heart of the quality systems for clinical laboratories. As I said before, ISO standards are non-obligatory. You do not have to comply with them unless they are memorialized in some type of law or regulation. So in the case of ISO 15189, you do not have to follow it in the United States unless your clinical lab is CAP accredited, and CAP being the College of American Pathologists. And many clinical labs are accredited by CAP, which means that those that are accredited by CAP do need to comply with ISO 15189. All CAP accredited labs should currently be in compliance with the 2012 update to this ISO standard, but there is going to be a transition to the 2022 update by the end of this year. So basically, by the end of this year, you should be in compliance, if you're CAP accredited, with the 2022 update to this ISO. Now, outside of the U.S., it's important to recognize, and we know that there are people from all over the world on this webinar. Other jurisdictions actually have standards that are required to be complied with for clinical laboratories, like France, Belgium, Hungary, Ireland, Lithuania, and Australia as well. Although Australia, it's kind of a blended situation where you do not need to comply unless you're receiving payment from Australia's Medicare system. So from a overall compliance perspective, kind of around the world, starting to look toward this ISO standard is going to be a good step forward. Even if you don't have to comply, actually moving in that direction can be beneficial. Now, on the other hand, ISO 13485, this is tied to the FDA, but this is not tied to the big rule that was vacated earlier this week. There's a separate regulation that will kick in on February 2nd, 2026. But this is for manufacturers of in vitro diagnostic devices, so test kits. Clinical labs, again, don't necessarily have to comply with this because, as we just saw from what the court determined on Monday, IVDs and LDTs are most definitely not the same thing. But still, there is this focus on quality management in designing, producing, and distributing devices. And what this ISO standard will do is it will harmonize the U.S. with the European Union, Canada, Australia, and Japan. So a large chunk of the first world already complies with this standard and already mandates compliance. So we will talk much more about these two standards as we move forward. All right. And this will be, I promise you don't have to hear me for too much longer. We'll get into the quality systems in a second. But just in terms of the effort to regulate laboratory developed tests, or for those of you outside of the US, these are often known as in-house devices or in-house assays. These are assays that are developed by a single clinical laboratory. In the US, the FDA has been trying for close to 30 years to regulate these with efforts starting out as early as the mid-90s. But we have seen a much more concerted effort the last 10 years or so with both guidance that the FDA has put forward, as well as a discussion paper, and congressional efforts to try to memorialize who would actually regulate these LDTs, with one set of representatives and senators trying to push for CMS to retain control through the VITAL Act, and then another set of legislators trying to push for FDA control through the VALID Act. We also saw it during COVID during President Trump's first administration that the Health and Human Services General Counsel actually put forth a memo outlining the key reasons why the FDA should not regulate LDTs. And a lot of that rationale was put forward by AMP in their fight against the FDA recently in Texas. So fast forward to last May, that final rule got published. Everyone was working feverishly to try to comply and get ready for stage one. And then on March 31st, 2025, or on Monday, the rule was vacated. So one thing that's really important for people to understand, and we end this slide with a question mark, is that determination is not final. That trial court made that determination. If the FDA so chooses, they still have another few months where they can appeal to the Fifth Circuit, and then eventually they could potentially appeal to the Supreme Court. Now, the Trump administration is as I'm sure many people have witnessed, is trying to really peel back the government, the federal government in particular, the regulations associated with it. So there is that question as to whether or not the FDA and the Department of Health and Human Services will actually appeal that decision, or they will just kind of let sleeping dogs lie and will allow basically that district court decision to stand and be that final decision. So stay tuned. It is most definitely still turbulent times in terms of how we are going to move forward and try to understand this area. But with that, I am going to turn it over to my colleague, Travis Schlappe, who is going to talk about how you can prepare for these turbulent times.
Okay, thank you, Michael. Let's go ahead and dive into quality now and how we can deal with the turbulence around us. So the FDA has many different regulations, all in 21 CFR Part 820. We're only going to cover a few of them that are focused on quality, that are important regardless of whether the new rule goes into effect or not. It looks like it's not, as Michael said, but these QMS or quality management systems are important for any clinical lab, especially if you need to comply with ISO 15189 as part of your CAP accreditation, or because you're an international clinical lab. So there's complaint handling, there's design controls, there's purchasing controls, there's CAPA, corrective and preventative action, and there is records requirements, all important quality practices that labs can implement to reduce errors and improve patient outcomes and potentially lower costs. So first, complaint handling. Develop standard operating procedures if you don't already have them for your complaint handling. Have an SOP for it. Have a complaint handling unit. Maintain detailed records, and then training staff on how to deal with complaint handling. I would assume that a lot of you are probably already doing this, but as a reminder, there are best practices and formal systems for complaint handling if you need guidance from 21 CFR 820. Next, we go into design controls. What are design controls? There's various components, as you see on the diagram on the right. You start with user needs. What do my patients, what do my providers, what do my hospitals want? Go into design inputs, then a design process, then a design output, and then you're going to verify. You see that going to the left there, verify that the design outputs meet your design inputs. And if they do, then the next stage is you have a final medical device that you validate meets the user needs. At each stage, you see there are review stages to make sure that each process is happening according to the specs and according to plan. So we'll start with design inputs. Not going to cover all of the components of design controls, but just highlight the 3 or 4 most important ones. One of them being design inputs. You probably use other, you're probably using design inputs right now. You just call them differently. You call it a design requirement or a product requirement, maybe a design input requirement. ISO, FDA, call them design inputs. It's all the same thing, but there are 3 kind of categories of design inputs. Functional inputs. So what does the device do? I test saliva for COVID. What are its inputs and outputs, like the sample input? What are the results outputting? Performance, how fast does it go? What's the turnaround time? How accurate is it? Sensitivity, specificity. What is its use environment? For most of us here, the use environment is a clinical lab. What kind of shocks or vibrations should it be able to withstand? What are the limits of its operation? And then interface, what are we connecting it to? Computers, Bluetooth, other things. What compatibility with other devices does it need? These are all examples of design inputs that would go into the design of your laboratory test. So I made up an example here to help us understand translating user needs to design inputs. Let's pretend we had a 3-in-1 molecular test to detect COVID, flu, A and B from saliva. And this was a new LDT that I had in my lab, and I wanted to follow best practices in quality to design this test and reduce my errors and improve the accuracy and outcomes. I would start with my user needs. Okay, so for example, in the second row there, I want to detect low concentrations of the virus. That's my need. Is I need a sensitive test that detects low concentration of the virus. That's why we're coming to the lab and not just doing a rapid lateral flow assay. Now, a design input would be the assay will have an LOD of 100 copies per milliliter or lower. So these design inputs, you'll notice they need to be specific, objective, quantifiable, so that you, in your design process, can actually verify that you've met the input or have not met the input. Don't be too specific. You see on the bottom there, don't over-specify. Don't say the device will weigh exactly 2 pounds. Don't say the device will have a sensitivity of 94%, because then that's going to clog up your design process. You may hit a sensitivity of 98%, and you would fail that design input requirement of exactly 94. So say 94 or above, right? LOD of 100 copies per milliliter or lower. Give yourself a range and some flexibility when you make your design inputs, so that throughout your design process you can meet them without having to redo the design input process. Let's go to the next slide here. What is the scope for design inputs? You really could make it as large or as small, as you want. It's up to the lab. It depends on the complexity of the test. It depends on the risk of the test. Here's some examples. Once again, best practices that the FDA gives for your scope of the design input: biocompatibility, toxicity, safety, like I said, performance, labeling and packaging. There's a whole list here of things that you could make design inputs for. I'll go over one example of how to go from vague and broad into narrow on a design input. So if you look at the figure in the bottom right, we start with the device will be AC powered. Okay, that's helpful, but not specific, quantifiable, objective. And so I'm going to X that out, and I'm going to go to the next stage. Well, I want my device to be operable from AC power in North America, Europe, and Japan. Once again, helpful, but not quite specific enough. Typically, systems run at 120 volts, but many systems are designed to operate over a range. And if it's a critical device, then you probably want around 95 to 132 volts to account for brownouts or other potential problems that happen. So I will go to this third level, and I will say my design input is that the instrument is operable from 95 to 132 volts. This gives me a good range. I can meet that spec. I can specification. I can verify it and move on to the next stage. So next, design outputs. I have my inputs. I do my design process. Now I have outputs that verify the inputs. So examples could be assembly drawings, production and process specs. It could be a piece of device software code that's analyzing some fluorescent signals, for example, and then verifying that I met the design inputs. It could be packaging and labeling specifications, the results of a risk analysis, biocompatibility test results, verification activities. These are all outputs that verify my inputs are being met. So once again, example here with my 3-in-1 molecular saliva test. Let's go to the third row, my design input, as I want this to be high throughput during flu season. That's a user need. I translate that to a design input or a product requirement, which is that the device or my instrument will process 96 or more samples in less than 8 hours. I'm just making up numbers here. You may have more ambitious goals than that. And then the output could be a schematic of your throughput and some documentation showing over multiple days, multiple runs, that your device's runtime was adequate and met this specification of 96 or more in less than 8. Another user need could be that it's easy to use for your lab staff in a clinical lab setting. So your design input, okay, the workflow will have less than 3 steps and require less than 12 hours of operator training. We're getting specific. We're getting quantifiable here. I can actually verify that I've met this by making a finalized SOP and doing some human factors engineering studies, and then showing those results as my design output. So those are some examples of design outputs to verify your design inputs. Next, we go to verification. What does that look like in a clinical lab? Well, it could be a failure modes and effects analysis, a thermal analysis of your assembly. It could be a fault tree analysis, package integrity tests, comparing your lab test to a previous lab test that has an established history of successful use. So these are things that you can do to verify that your lab test is meeting the specifications that you set forth. In our example of the 3-in-1 molecular test, let's go to the third row. I had a design input of a limit of quantification of less than 500 copies per mil. My design was a qPCR instrument with a 96 well plate, and then my output could have been a TPV, test performance verification 2.2, where I did an LOQ study and I demonstrated that I hit the LOQ of less than 500 copies per mil. That would be my verification that I met my acceptance criteria. Other examples are FMEA tables, traceability matrix, or sign-off documentation by reviewers, by management, that, hey, we're just verifying, we sign off that we met these design inputs. Validation is the next stage where verification is asking the question, does the device perform well? And does it meet the specifications that we set? Validation, on the other hand, is asking, does my device meet user needs and the intended use of the lab test? And so ways to validate that we met user needs is, the easiest, well, not easiest, a lot of time and money, but the most simple example is clinical trials, validating that your test meets the user needs and has the appropriate sensitivities and specificities for patient samples. You can provide historical evidence that similar designs and materials are clinically safe, analysis and inspection methods, compile relevant scientific literature. As I mentioned before, human factors engineering or usability studies, validating that we're meeting the user needs of the lab staff, making it easy to use, easy to operate, things like that. Okay, so validation in our 3-in-1 molecular test example, if my user need is easy to use in a clinical lab, then my validation could be a usability study showing those with less than 12 hours training, and the instructions for use can operate with a less than 1% error rate in less than 3 steps. Now, when you do this usability study, don't, cheat the system and use your most skilled lab technicians. Use your average lab technician. We need to simulate the actual usage of these lab tests and not just use our best ones to pass the validation study. Another part of validation could be a feedback survey of lab personnel showing that 85% or more rate the test as easy to use or very easy to use. On accuracy, those are the simple example of doing validation studies demonstrating more than 95% sensitivity for COVID. Could be your own clinical study, could be comparing to a gold standard or predicate device. And then for specificity, similar thing, doing a study demonstrating 98% specificity or larger. Notice those greater than or equal to signs. I'm not restricting myself to exactly 98%, but anything above 98% will pass my validation. Okay, so on to International Standards Organization, their quality standards for labs and IVDs. As Michael mentioned, 15189 is voluntary for clinical labs. Unless you're using CAP as the accrediting body, then you're likely already meeting these requirements and it's required for you. ISO 13485 is more on the IVD manufacturer side. And as Michael mentioned, there'll be a new rule for the manufacturers going into effect on February of 2026. Overall, though, if you're an international lab, you're probably needing to comply with 15189, and potentially the whole direction of laboratory medicine and diagnostic tests is moving towards quality. And at some point, ISO 13485, those processes are best practices, and they're good to have in your lab. I'll show some evidence in a few slides of how quality can improve your lab error rates, turnaround times, and things like that. When we, I won't say comply, because you don't have to, right? Unless you're CAP accredited of 15189, but I'll say adhere and implement these practices can improve the quality of your lab and therefore the outcomes of your lab. So ISO 13485, this is the international standard for IVD manufacturers. It emphasizes more on risk management and leadership roles and management than the FDA's version. So quickly there, the key elements of 13485, which are good elements not just for lab medicine, but for any process, any business or technical engineering scientific process. Plan, they call it plan, do, check, act. So plan what your process is, how you're going to do it, whether it's purchasing, suppliers, designing a product, validating, plan, then do it. Okay, then check, do your measurements, your analysis, your improvements, and then act based on your review stage. So after you've checked, you review, you find some things to improve on, and then you start the cycle again. Plan the new process, do it, check, and then act. Plan, do, check, act. And you would document this in things like a quality manual, medical device file, document control. Don't worry about the names of these documents. This is more if you are an IVD manufacturer, you can call them other things, but these are good names to use so that you can implement quality into your clinical lab. The 15189 standard has a variety of components that are very similar to the 13485 standard. There's management requirements. You'll see corrective action in there, preventative action, records control, management review, document control, quality management system. So similar stuff, and it's adapted to clinical labs rather than IVD manufacturers. You see the quality system essentials there on the right, where your process management, your personnel, facilities, and safety. They have standards, explanations for how to do all of these things to have the highest quality. Okay, so as we've said, if you're CAP accredited, you're likely already complying with CAP 18589 based on CLIA law or what you're doing. The inspections happen every year on an internal basis, like internal audits every year. External audits are on a 2-year accreditation cycle. On the right shows a figure of the risks and benefits, or let's call it the costs and benefits. We know it's going to cost money for staff and time to do this, documentation, training. The question is, do the benefits outweigh the costs? Well, that's going to depend on your lab and what you deem. I do have a few sources in the next slide of what other labs experienced on the benefits and costs of implementing these quality systems into their labs. But the benefits are potential cost savings. You have less errors, and errors can be very costly. Reductions in inspection deficiencies, staff engagement and morale goes up. And you build a culture of prevention and seeing errors before they happen, rather than a culture of fixing mistakes and fixing errors after they happen. So is there any evidence that the quality is actually a positive outcome for a lab? And so most of these papers come from labs implementing ISO 15189 systems into their lab. One paper found that labs that had 15189 had about a 0.7% error rate compared to 1.4%. So it doubled for labs that did not have those quality systems. And they also did a before and after measurement. Labs that were not adhering to 15189 had a 1.3 error percent. After they got accredited with ISO 15189, it cut in almost half to 0.7%. There was a study with some hospital labs. This was done in Iran. Most of these studies are actually international, Austria, Iran, the labs that are complying with 151, 89, or 13485. They had a 13% reduction in the cost per test. It went from $1.80 to $1.57 on the test that they were studying. And then also a Lean Six Sigma, which is another quality approach, eliminated 3 hours in pre-analytical workflow, cut the average turnaround time by 13% for a lab. When management emphasizes quality, so if you're lab manager and if you're really the champion of quality, you can see significant reductions in turnaround time, specimen rejection rates, significant increases in clinician satisfaction rates, and then decreases in nonconformities. So you likely will see cost savings because you're going to minimize the need for multiple tests, prevent inaccurate diagnoses, and streamline treatment protocols. You also will likely see positive laboratory incomes, reducing errors, more effective operations, and you'll have sustainable attainment of quality objectives. You're supposed to be doing QC and QA. You already are doing QC and QA, but the documentation, the management of it may be disorganized or haphazard. And when you start applying these best practices from the FDA or from ISO into quality manuals, quality management systems, it streamlines things. It makes it more sustainable and less stressful to do the QA and QC. So how do we implement these best practices? There's education and training. FDA has a whole website, Division of Industry and Consumer Education, of how to implement a lot of these quality practices. I recommend starting with a gap analysis. What documents do we currently have? You likely already have a lot of documents of verification activities, validation activities, things like that. And then what are we doing but not documenting? So we're probably doing design inputs, but are those living in the head of one of our employees instead of documented somewhere so that when we have new employees and new lab personnel, we know what the design inputs are, outputs, verified, validation activities, all of those things. Do we have a quality management system, or once again, is it just kind of living in someone's head? Do we have pilot projects? And then there are documentation tools. You can purchase software subscriptions or use Excel and files and Word documents to organize, however fits your lab. So there are some resources from, like I mentioned, FDA, CLIA, ACLA, ADLM. They all have resources on improving quality in the lab. There's consultants and third-party quality organizations. Start with just training on QMS and design controls, and possibly engage with external resources for guidance and support. So here are some of the resources used in preparing this presentation. FDA has guidance documents for design controls, walking you through how to do them and giving examples of how to do them. There's quality system information for pre-market application reviews. If you're ever thinking of taking your LDT and moving it, through the FDA approval process and getting a PMA, quality is a big deal for any pre-market application that you do. So important to get those quality systems started now. It's going to make your PMA process go smooth. Here's our contact information if you want to reach out with any questions after the seminar or about the diagnostics program at ASU. And we will now move on into the Q&A session. So any questions, go ahead and put them in the question box.
All right. Travis, thank you very much. That was great. Tons of helpful information. So we have a few questions that have come in. The first one being, and I'm not sure if you know the answer to this one, but I know that I don't. So I'll at least ask you, what are the recommended compliant LIMS and ERP softwares for CLIA labs?
I don't know for sure the answer to that. I don't think there is one that is like, this is the only one that's compliant. I think there are several that could be compliant, but I'm not going to talk too much on this because I'm not actually sure. I don't want to guide you in the wrong direction. I don't think there's a single one, I guess is my answer. There's probably multiple.
All right. No, that's fair. Thank you.
I mean, it probably depends on how you use it. If you're using it in compliant ways, then then it's likely fine. If you pick one and the software you choose is not abiding by the regulations, then that software is not going to be fine.
That makes sense. All right. So next, so do clinical labs really need to worry about ISO 13485? What do you think?
I would say right now, no, based on Monday's decision, no. So before Monday, yes, because in February, ISO 13485 was going to be a part of IVDs, which clinical lab, LDTs, were they going to be part of IVDs? So as of right now, I would say no, focus more on ISO 15189. However, as Michael said at the beginning, pay attention to the news. You know, if the FDA appeals, if this thing goes back to where it was, then yes, ISO 13485 will be important.
All right. Thank you for that. So next one. So the future of these regulations and new regulations, and this one I can take. What is happening in the courts with the Trump administration and Congress? Is there any chance these items are actually, or these regulations are going to go into effect? There's always a chance. I cannot give you a yes or no answer for sure. So obviously May 6th is, gosh, just about exactly a month away. As I said in the intro, we are looking at, I think it's 60 days from that that March 31st decision that FDA and Department of Health and Human Services has to appeal to the Fifth Circuit. Thereafter, there could be kind of quite a long appellate process that happens and could eventually make it up to the U.S. Supreme Court for a final determination. There has been, in addition to that, the entire judicial process, there could be efforts in Congress. I don't see that happening. Again, that's just my personal opinion. There's plenty of things that Congress has going on, and I don't know that anyone's going to spend the political capital on trying to move this over, to move LDT regulations over to the FDA officially. Because what the court came out and said is the FDA doesn't have the authority to regulate LDTs. The FDA could have authority if Congress basically gave it authority. So basically FDA overstepped its bounds. So Congress could still step in and make that determination. President Trump would have to sign any bill that came out of Congress. And then, as I mentioned from the top, there's still that overarching issue with respect to the Trump administration just taking an anti-regulation position across the board. So you've for those of you again who have paid attention to the news here in the U.S., Department of Health and Human Services just announced they're going to let go of 10,000 people. Even before that announcement, FDA would have had to hire additional people in order to try to enforce this rule. So it would even if the FDA had this authority, it would be really hard to enforce these rules anyway. So long story short, my guess is on May 6th, these rules will not go into effect, but that does not mean that again, to the entire point of this presentation, looking for strategic approaches to quality is not a great idea, because there is, as Travis mentioned, there's some great benefits to that, both in terms of operations as well as money. Let's see here. And then, so we have one last question. And that is, this one will be a Travis question. So for ISO 15189, how often is an internal audit necessary? And how often are external audits necessary to maintain accreditation?
Yeah, sure. The internal audit is annually, so that's just within your organization. You do an audit, document it, make sure that's all good there, and then the external audit is usually, at least for CAP, every 2 years. There also may be surveillance audits that are quicker and not full audits, maybe just checking one part of your processes. They may do a surveillance audit and just check CAPA, for example, every 6 to 12 months. But the full audit is once every 2 years, and then internally once a year.
All right. So just real quick, a quick follow-up. What kind of documentation is going to be necessary for that internal audit? Do you know?
Yeah, so the standard will tell you what the documentation that you need is, but it'll likely be checklists. At least I know the FDA uses checklists of all the items that you are checking or that they are checking. Sign-offs from management. It could be some data of, for example, stability testing or something like that. So it depends on what your SOP says of how you do your audit. You write the SOP. This is how we're going to do our audit. And then you compile those documents and data. Make sure your SOP complies with the standard.
Excellent advice. Excellent. Well, that is it for the questions. I will turn it over to Mara Aspinall. Oh, Mara, I think you're muted.
Sorry about that. I wanted to say mute earlier. I hope you enjoyed and found this presentation very useful. Clearly, given the decision on Monday, we wanted to bring you not only the implications of the decision, but what are the best practices and quality in each of your labs, regardless of the change in the LDT regulations. So what I wanted to do was thank Thermo Fisher, thank our two great presenters, Michael Donovan and Travis Schlappe, for this key information and answering so many of your questions. If you have additional questions on the content or our program on biomedical diagnostics, please follow up with us. And, in the midst of our time here with a lot of focus on healthcare, I can't emphasize enough the importance of diagnostics and the importance of each of your labs. I really call it the glue that brings the healthcare environment together, whether you're doing research or, for virtually all of you, clinical work. The quality of what you do represents you and, frankly, represents the whole industry. So we hope that this is useful to not only work within the regulations in the US and around the world, but frankly, just bring better quality and continue to have that quality in each of your own labs. So with that, I say thank you. We look forward to keeping in touch and doing other materials in the future. I'm going to go forward from there. Is there, I guess there one other question about information, but again, please reach out individually if you have individual questions. Thank you.
Okay, I want to say a thank you again to our presenters for their time today and their important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Thank you. Take care, everyone. Goodbye.